Dosage compensation in Drosophila melanogaster occurs via an upregulation of the X chromosome and is mediated by a compound called the MSL complex. The MSL complex binds to high affinity sites on the X chromosome to facilitate dosage compensation. These HAS are highly interconnected, and the long-range interactions between HAS facilitate spreading of the MSL complex. ROX genes, such as ROX1 and ROX2, are located on the X chromosome and serve as a marker to distinguish the X chromosome from other chromosomes, such as autosomes. The RNA products of the ROX genes are only stable when bound to the MSL complex and increase the affinity of the complex for the X chromosome binding sites. MSL is made out of multiple components. MOF and MSL3 interact with MSL1 and MSL2. MLE, an RNA helicase at the high affinity sites, causes ROX1 and 2 RNA to be incorporated into the complex, stabilizing the RNA. There are two theories for the mechanism of MSL spreading. First is the idea that high MSL concentration at HA sites causes less selective binding of MSL to surrounding lower affinity sites. Although the non-HA sites have lower affinity binding to MSL, there still can be a stable interaction between the complex and the DNA due to high concentrations of MSL nearby. Secondly, modifications to the DNA caused by the MSL complex may allow it to spread across the chromosome. When MSL binds to the DNA, it causes acetylation of histone protein H4K16AC, which then allows MSL to access low affinity binding sites. This is supported by the fact that MOF causes acetylation in nearby nucleosomes. Dosage compensation is targeted to active gene segments that have been transcribed. To identify these segments, histone H3K36ME acts as a marker, which may be recognized and bound by MSL3. Two additional proteins that may be involved are H4K20ME1 and H4K16AC. The HAT domain of MOF catalyzes the acetylation of H4K16AC, while MSL1 and MSL3 increase the enzymatic effectiveness of HAT. RNA polymerase II transcribes genes to their end more effectively at the X chromosome, and this facilitates the upregulation of the X chromosome. This is due to the histone acetylation mediated by MSL, which allows for more effective polymerase movement for better transcription. Additionally, transcription factors are better able to access gene regions which are acetylated.